protein purification. Protein purification means to purify the protein and non-protein parts of a cell. Purpose of protein purification is for the characterization of the function, structure and interactions of proteins of protein of interest. Steps used in the purification of proteins are: number 1, choose source of protein. For the interest of protein, first the cells are broken down or cell lysis is done. Two methods are used for cell lysis, that is physical method in which the external force is used to break the cell membrane, while the second method is the chemical method or reagent based method in which the lysis buffer is used for breaking the cell membrane. Next, after cell lysis, the crude lysate is taken and then filtered or centrifuged to remove the cell debris, and the protein of interest is present in the supernatant, so supernatant is separated. Second is to solubilize the proteins. For solubilization, proteins are get into the solution. If the proteins are present in the cytosol, then we need to break open the cell. Different methods are used to open the cell. For that, if the cell is animal cell, then the osmotic lysis is used. For this purpose, the cell is put into the hypotonic solution. If the cell is with the cell wall, means that the cell of bacteria or plants, then the lysozyme is used, or detergent are used, or organic solvents are used. These three methods are used for the bacterial or plant cells to open its cell. Number third step is to stabilize the proteins. For stabilization purpose, pH, temperature, inhibition of proteases and the retardation of microbes. For retardation of microbes, the sodium azide is used. By using all these four, the proteins are stabilized. So these are the three steps used for the protein purification. That is to choose the source of protein, solubilize the proteins and then stabilize the proteins. Protein sequencing. Three methods are used for the sequencing of proteins. Number 1, N-terminal sequencing, that is the edman degradation technique. Second is the C-terminal sequencing, and the third is production from DNA sequence. First, we will discuss the N-terminal sequencing. N-terminal sequencing are the edman degradation technique that is developed by Pierre Edman. In edman degradation, the amino acid residues are labeled and then cleaved from the peptides. Three steps are involved in the edman degradation process, that is coupling, cyclization. Number third step is the conversion. Procedure used by the edman during the degradation of protein, that is number 1, protein sequenator is used, which is a machine consists on the liquid phase sequenator and a cylindrical cup which is known as the reaction vessel. Number second, protein samples or peptides are immobilized in the reaction vessel. Then the coupling of these peptides or protein samples are carried out with the phenyl thiocyanate reagent. Phenyl thiocyanate or PITC reagent is used for the coupling purpose. Number fourth, during the reagents, that is the phenyl isothiocyanate and the extracting solvents are passed over the immobilized film of proteins or peptides. Number second, the cyclization process in which each cycle releases one amino acid from the protein or peptides interminus. Edman degradation principle is also based on the phenomena in which during one cycle one amino acid is released without the breaking of peptide bonds between the other amino acids. Then conversion takes place in which the high pressure liquid chromatography or HPLC is used to identify the released amino acids, and the process is repeated for the whole polypeptides until the entire measurable sequence is established. Look here, that is the diagrammatic process of edman degradation, in which this is the protein sample and that is the labeled amino acid which is to be cleaved. For coupling purpose, phenyl isothiocyanate reagent is used, that is the coupled protein or peptides. Then the HPLC is carried out and at the end the released amino acid is collected. एडमन डिग्रेडेशन प्रोसेस और इन टर्मिनल सीक्वेंसिंग के अंदर प्रोटीन सीक्वेनेटर यूज़ किया जाता है. इस प्रोटीन सीक्वेनेटर के अंदर लिक्विड फेज सीक्वेनेटर होता है और उसके अंदर सिलेंड्रिकल कप लगा होता है. सबसे पहले प्रोटीन सैंपल्स को इस सिलेंड्रिकल कप या रिएक्शन वेसल के ऊपर एक टिन फिल्म के फॉर्म में इमोबिलाइज कर लिया जाता है. फिर उसके बाद इसके अंदर फिनाइल आइसोथियोसाइनेट रिएजेंट डाल दिया जाता है जो इसके ऊपर मूवमेंट करता है. हर साइकिल के अंदर एक ही अमाइनो एसिड रिलीज़ होता है और यही रिलीज्ड अमाइनो एसिड जो है इसको फिर थ्रू HPLC और हाई प्रेशर लिक्विड क्रोमेटोग्राफी टेक्निक के ज़रिए से आइडेंटिफाई किया जाता है. इसके बाद इस प्रोसेस को रिपीट किया जाता है उस टाइम तक जब तक कि होल जो पॉलीपेप्टाइड चेन हमारे पास है उसके जितने भी अमाइनो एसिड्स हमने आइसोलेट करने हैं जब तक कि वो सारे के सारे आइसोलेट हो जाएं. Second is the C-terminal sequencing. steps involved in the C-terminal sequencing are: take the protein and the solution, then carboxypeptidases are added to that protein solution, take samples from that protein solution at regular intervals and make a plot of amino acid concentration against the time to determine the terminal amino acids. Third is reduction from DNA sequence. Protein sequences can be determined indirectly from the messenger RNA or from an organism that have no intron, for example prokaryotes. Then sequence a short section that is almost 15 amino acid long. Number third, primer is designed from the amino acid sequence. Then gene is amplified and gene sequences is carried out, and at the last to determine the amino acid sequence of the protein. So these were the three methods used for protein sequencing. They were N-terminal sequencing, C-terminal sequencing, and the production from DNA sequence.